So hi, good morning again, everybody. So my name is Kylie from ABCAM. So it's really my pleasure to spend an hour with you guys. And welcome. Thank you for joining us today. So I truly appreciate you taking the time to be here, sharing the knowledge and your research passion here. So in this 1 hour, we will explore the different types of antibodies, take a closer look on how ABCAM maintain the quality and our innovation to make sure that the consistency of our antibody product that really plays a crucial role in your experiments and in advancing scientific discovery moving to the future. So before we dive into science behind an antibody types, so I like to take a step back and highlight an important foundation of all research will be reproducibility. So understanding why reproducible results matters, set the stage for why antibody quality and type of antibody selections is so essential for proteomic research. So let's take a closer look at just how big the problem really is when the result of one paper is not reproducible and the downstream impact. So the scientific community refer to this as the research reproducibility crisis. And a few examples in this slide shows on highlight on how deeply it can affect scientific progress. So first, there's a paper in 2017 by Anderson. So uncovered that some of the most widely used antibody in the literature, those trusted by hundreds of researchers for decades, were consistently binding to the wrong protein. Even more concerning, the target protein wasn't present at all in the breast cancer tissue being studied. So this means that 2 decades of research were influenced by incorrect assumption due to the result with the data they have and sending countless projects downstream in the wrong path, and soon paper retraction, right? So in 2013, there's another research found that antibody believed to be specific for CDK1, one of the target, also recognize a completely different protein. So this antibody have already been used in over 200 published papers. So this isn't a small error, you know, like it ripples the spread across the scientific literature, causing the future studies that has been built on the result. So this is where the real downstream consequence come in, right, that you can imagine. So once an incorrect science being published in the literature and misguide the future project, which also cause paper retraction, which will affect reputation and integrity of the field, and of course research funding. So over many years, as researchers pursue conclusion built on the unreliable data, right? So it really affects the downstream experiment that the other researcher trying to reproduce. So next, okay. So then another research paper also did highlight that the financial burden of this problem. So it emphasized that the dramatically improve of reproducibility with the protein binding must be defined by their sequence and manufacture consistency, right? Otherwise, we risk wasting enormous amount of resources in terms of like time, effort, funding, which is something that's not easy to get nowadays, right? So there's also another article paper has addressed that even 50%, they do a research with a pool of 1,500 scientists, right? These 1,500 of scientists are not even able to repeat their own design experiment. And 36%, more than 1/3, is due to poor reagents. So in short, the choice of reagent, like type of antibodies, and also where it comes from, the supplier, is very important for the proteomic research work. So at ABCAM, we committed to developing antibody with better specificity, performance, and consistency to ensure a reproducible result in the research area. Apart from the type of antibody, Quality control is also a very crucial step to maintain antibody performance, which then highly linked to, again, the specificity, consistency, and reproducibility. So currently, ABCAM has over 65,000 of antibodies for more than 25,000 targets, which you can already imagine the extensive work we have committed to every single of our product antibody, from target selection to QC stage and deliver out to you or the researcher globally. So let's walk through the type of antibodies, how it's being produced, manufactured, which definitely will impact their performance on your research work. So first, we look into polyclonal antibodies, which has been way long existing in our market. The production typically actually start with immunizing an animal with the target antigen. Then to stimulate an immune response, come with a production of an antigen-specific antibody by the animal's B cell, right? So this immunization is repeated several weeks to increase the affinity of antigen-specific antibodies within this animal, and then it results the antibody can be isolated from the serum, so using affinity purification. So we isolate it through affinity purification. So you can see that polyclonal antibody are a heterogeneous mix of antibodies derived from the immuno response with a multiple B cells. So you can, like we highlight in different colors, but they are still recognizing the same antigen, just at different epitope, right? So by binding to several different epitope, so polyclonal, one of the benefit is they can produce a strong signal. So imagine that then after that we will bind the secondary antibody, which cause some strong signal amplification on your result. But please be mindful that at the same time, polyclonal can have a higher chance of cross-reactivity with homologous protein or isoform, as might have a similar epitope binding site because some of the protein are pretty similar. So result in a poorer specificity, and it will cause you an inconsistent result. So maybe this batch, so you look at your left-hand side. So maybe you buy the lot 1, you have antibodies, you see the purple one. Maybe in the lot 2, you don't have the epitope, the purple epitope. So maybe the purple epitope actually gives you the best binding site, right? The best binding. So it will have a better result in lot 1 and lot 3, but not in lot 2. So that when you buy the antibody, the performance pretty much is different every time. So additionally, because of the immune response, so you cannot expect the animal that we immunate to have exactly the same immune response every time. So that's the reason why it causes the difference of lot 1, lot 2, and lot 3. So for example, like human, when you react with COVID-19, with the recent, not very recent, but a few years ago, the pandemic. So some of the people have a very light symptom, some have very severe symptoms, some is like fertile symptom. So everyone immune system is different, right? So this is pretty much similar to the animals. So due to this feature, they are rather limited in supply in long term. So not just inconsistent result, but you might also not getting the same product in long term. So if, for example, touch wood that the animal die, so you're gonna find another animal. So it's the batch will be totally different again. So that's the disadvantage of polyclonal antibodies. So to improve the producibility, monoclonal antibody come in this place. Same process again, that we inject an antigen and create an immune response in the animal. And then we screen through all the B cell and find out that maybe this clone is performing the best. So we will fuse the B cell into a hybridoma cell, pretty much equivalent to cancer cell, for a long-term generation of identical monoclonal antibody in one single tube. So as monoclonal antibody come from a single B cell clone, so it only recognizes a single epitope per antigen, right? Therefore, it's less likely to have cross-reactivity, but at the same time, it will not produce a very strong signal in compared to polyclonal, but it's very specific, right? So there's a minimal batch-to-batch variation. As you can see, every batch will be the same. However, one of the downside to hybridoma antibody generation is that they may be vulnerable to stability issues because end of the day, it's still a cancer cell line. So when we passage it for a certain time period, it might result in some loss of epitope recognition due to mutations because of the feature of a cancer cell. So in conclusion, it's also not a long-term supply for researcher. So at ABCAM, we have the largest and fastest growing collection of rabbit monoclonal antibody using our patented RabMap and technology. So it has a higher affinity, higher specificity, and consistency to diverse and novel target. So remember the reproducibility crisis mentioned during the introduction. So this product is an enhancement above the polyclonal to address the challenge, which polyclonal, again, have a limited long-term supply and high risk of cross-reactivity, which leads to inconsistent result. So in ABCAM, RabMap antibody also undergoes extensive application validation before releasing them to ensure the lot-to-lot consistency, which I will explain more in the second section. So at the floor, you might actually wondering like why so big thing about like rabbit, why they have a higher affinity and specificity for the target antigen compared to the other host animal. So this is because rabbit has a longer CDR length, as highlighted here, means that there is more diverse epitope recognition. So rabbit generally have a better immune response in compared to other animal and have more successful at generating a very strong immune response to those like small molecule and peptides. So another plus point is rabbit also have a larger spleen. So the source of the B cells is higher. So a wider variety of antibody clones we can actually generate out from the rabbit and take a full screening. And they actually have also higher affinity as shown here. So like, for example, rabbit, they have like pico, but compared to other, it's only at nanostrain range. So in a simple term, like a sticky tape concept, the longer, which is the longer CDR region, it has, so it's stronger, it stick on the target protein. That means a higher affinity to the epitope on the target that we want, and it's harder to take off. So this is a comparison table as a summary. Compared to mice and other rodent, rabbits basically, again, when inject a protein target to the host, which is rabbit, have a better immune response with more B cell variation to produce the antibodies that's really recognize several epitopes. Furthermore, again, with a better immune response from the rabbit, it reacts a broader range of the compound. It's more easy for our productions. And again, higher affinity as shown here. Yeah, so with all these features, it highlights the performance, important performance, especially for IHC, ICC, and IP, which really require all these like stronger affinities and higher specificity in this application. So it's not as strong, not always, for mouse. So you can see the limitation and comparison among these two host species. So just to repeat on what we have mentioned for our rep map, it's lot-to-lot consistency. But the downside that mentioned in a couple of slides before is because it's a hybridoma antibody generation technology, right? So it's not very stable due to the cancer cell feature. So still not a long-term supply. So across the timeline, there will lost some epitope recognizations. So how we can improve further? The answer is we move into our recombinant monoclonal antibody technology, where we develop in vitro using synthetic genes. So after deriving the monoclonal antibody from the hybridoma cells, basically the antibody is sequenced. We will sequence the antibodies, sequence in DNA form, and then encoding the sequence and carefully control, allow optimized binding site and improve reproducibility over monoclonal antibody produced from the hybridoma, right? So once an encoding system is determined, so then we insert into the plasmid, transfect into the expression cell. Normally we will use HEK cells, and then we can ensure that all the translation and foldings come out from the cell culture and form our recombinant monoclonal antibodies. So therefore, the term recombinant really refers to the in vitro using synthetic gene compared to the traditional and monoclonal antibody and polyclonal antibody. So recombinant offer a long-term and secure supply with the least batch-to-batch variation. Since the antibody coding sequence is known and defined, right, it can further engineer. Sometimes we can manipulate with whatever intended we want to. So it is also very easily scalable in cell culture and can be used for high throughput manufacturing. So for example, actually, if not sure if you realize, some of the customer buying polyclonal, if we don't have stock and we will need to wait for new production, the new production basically need more than 3 months and depends on the animal reaction, maybe longer. So if we immunize one antigen to animal, right? So it takes like roughly 3 months for them to like build enough yield before we can purify it and then out to customer. But recombinant is different. So it's always in stock with us, depending on the popular target. So this slide further emphasizes the staining of different batch of recombinant antibodies, such as this is a PDL1, a very popular cancer target, providing consistent results. So first year, third year, or 5 year, or so on later, result will be exactly the same as today. So this is to further capitalize on the advantage of our recombinant antibody. So something interesting and innovation from ABCAM is we produce a multi-clonal antibody. Next slide. So recombinant multi-clonal antibody are a defined mixture of carefully selected individual recombinant monoclonal antibody designed to recognize the different epitope on the same antigen. So they are ideal solution for application where polyclonal antibody would traditionally be used. So remember the few slides before, polyclonal also bind at the antigen at different site, not for monoclonal, not for recombinant, but yes, for multi-clonal antibody with recombinant feature on it. Okay, the mix of antibody in this tube, this multi-clone tube, each clone is carefully controlled. So each of them are recombinant antibody with defined set of gene and encode the antibody with the desired characteristic. So this also ensures batch-to-batch consistency and reproducibility of the result. So our recombinant multi-clonal range has even get award by the Site Act in 2022. So example on when to use our recombinant multiclonal antibody would be when you are interested in detecting multiple post-translational modification or targeting different form of pan target. So it is also particularly useful when detecting low abundance protein because of their high signaling, like how polyclonal do. The recombinant antibodies can also be useful when you using the same antibody for multiple sample from multiple species as it offers a very broad epitope coverage. In fact, also multiclonal antibody can have better performance as compared to polyclonal. I give you an example. Look at your right side on the Western blot. Okay, so this is a recombinant multiclonal. So this is polyclonal. So polyclonal is not that specific enough to get a band in lane 2, but the recombinant is a yes. Okay, so hopefully it cleared off. So I just want to put this slide as a summary for one glance for you to emphasize the difference in epitope recognition style of each different type of antibody. Again, polyclonal is a mixture of antibodies that targeting different epitopes on an antigen, target antigen, but easily have a cross-reactivity on isoform of similar protein that have batch-to-batch variation, while RedMap produced by genetic DNA, right, will only recognize on one epitope. Multiclonal, every single clone is recombinant antibodies joined back together to produce exactly the same feature as polyclonal and have a long-term supply. So after we all get an idea and types of antibody in ABCAM portfolio, which also hopefully could help you in better selecting the right antibody for your research work. Next, I will move into the second part of today's webinar, explaining more about how ABCAM maintain our product quality, which also making an important role in maintaining the consistency. So RedMap and recombinant technology has already have an exceptional batch-to-batch consistency, sensitivity, and specificity, right? As mentioned in our section 1. So to take another level up, the newest initiation is the introduction of biophysical characterization of our antibodies. So using these methods such as DLS, LMS, and HPLC, we go beyond application performance and really get to know our antibody at a molecular level. So ABCAM is actually the first antibody reagent company to add this level of quality control in our manufacturing process. In terms of chemistry work, examine how molecular behave is equivalent to the molecular fingerprint identity. So we really know how this antibody is being fold, the purities through all this method. The sequence identity, sequence integrity, aggregation, whether they have aggregation and purity. So this is exactly, everybody have their own fingerprint. So this is similar to the antibody. Every antibody have their own fingerprint as the epitope sequencing is different. So once we have that molecular fingerprint of our antibody, we will absolute certainty that each batch of the product is identical, consistent performance is really guaranteed for all single type of antibodies. So we dive deep into this BBQC. So this is a LC-MS peak need to appear at a correct molecular weight peak for heavy and light chain, but which is not in this case. So the sequence identity is kind of wrong, incorrect. So look at another example of a chart below showing a lot of small peaks indicating the antibody is no longer in a Y shape that we want, instead of appearing in a very small fragments in the tube. So that might indicating there's some degradation might happen, maybe there's a mutation happen in the cell line that we will need to go back and really investigate what happened in our cell culture. So next will be HPLC, used to check aggregation due to storage condition, maybe the solvent composition of the antibody, and quantify if there's any impurity, which then notify our team that this patch need to implement some purification step to remove impurities before out to the customers. So another benefit of BPQC, it will be faster in our QC stage compared to application testing, like validating with individual application on the bench. Like imagine you will spend time for IHC here, and then spend time for flow cyto here. But this one, with BPQC, as we already know how it flow and their identity impurity, we will ensure that is what we want, right? Quick and easy with all this method being used. So in this slide, just to show that with BPQC, ensure lot-to-lot consistency of our manufacturing line. So different lot appear in our mass spec with a consistency of light chain and heavy chain in single peak. In HPLC, consistent purity, no aggregation, no contaminations. Here shows an example of two light chain, peak found in our MS, result at our QC stage. So immediate action for our team is to resequence it with the correct sequence and ensure that the correct translation into the antibody protein that we want for this app ID. So this is an example of like before the solution and after the solution, like there is a stronger staining, like even in IF, like really, really strong staining, right? So this is a snapshot for our BPQC, where ABCAM is proud to claim that we are the top among other suppliers. So apart from BPQC, another key highlights on our quality control is that we use knockout model. We use CRISPR-9 in-house to validate our antibody on Western blot. So information generally will be attached on our website, and you are worry-free to work on all these validated antibody to kind of explain further in another slides here. So look at example of these three examples. So this is a recombinant red map antibody. So based on this result, we are really seeing single band, no band in knockout, band in wild type is binding on whatever on the target that we want, PKC. So next we'll be looking at these monoclonal antibodies. They are still binding on the target that we want, but there is a weird band happening here. So this is some cross-reactivity and off-target happening. So what ABCAM will do, will remove this product and publish on our website, so you have no longer available. That's where I'm saying that monoclonal antibody and polyclonal antibody, there is not really a long-term supply. So another example will be this polyclonal antibody. It's totally off target too. There's even a signal and knockout sample. It's not something that we want. It's a wrong target. So this is how we do the knockout validation on our antibodies. So again, we are so obsessed with validation in an era where the reproducibility of science is in crisis. So we believe it's essential to provide reagents that are high performing, reliable, importantly consistent. So validation in application and sample is the most relevant to your research work. So some antibody work in some antibody work in Western blot, but some don't really work in IHC and etc. So we have already pre-validate the application for our customers. So we characterize and validate our antibody using, as you can see here, the covering most of the standard applications such as flow cyto, such as cytochemistry, IHC, which are typical for all our discovery programs and research. We are constantly expanding the variety of validation to include what is most important and relevant for you to able to select the right antibody quicker. So this is a screenshot I took from ABCAM website, where we actually share, we are very transparent. We share our application and validation data. As you can see here, we have IHC, IF, Western blot, probably immunoprecipitation here. All that's with proper descriptions for our researcher informations. So also on product range, application range. So this is also an example, a table to illustrate that what sample and what application we have already tested for our customer or researcher. Like this particular antibody is not work in flow cyto.
Faham ke? Too complicated. Saya dah masuk dah.
No worries, I will explain later and take questions later. So this is not really working for flow cyto, but only work in human sample and ICC and IF, only if the sample is human, right? So all this information are covered in our 12-month guarantee once you receive your product from Apical. If you have an application, for example, here, I will say like, for example, ELISA, you want to use this antibody to build your own assay or maybe work on a cat sample that is not validated here. We can offer something like trial program. So testing the antibody for free, subject to our investigation with our ABCAM technical team. So just send in your inquiry to Apical. They will support you locally in the process. Just don't be shy to ask. So to highlight our product promise, 12-month guarantee upon your product receivable, right? So if you keep in the right condition and work according to the data sheet, we will help you to, we also have a technical support to help you troubleshooting, also give you some advice for your experiment design. And if it doesn't work, we will give you a one-to-one placement. So this is an app trial program that I mentioned before. If you have an application that is not validated by us, feel free to approach Apical. We will bring you through the process and you can test the antibody for free. So in case you are curious on how to search the right antibody for your target of interest, you know, when you like search for P53, those like famous target for cancer, you will see more than hundreds of antibody products will appear. So which one is the best for you, right? We have built filter. Oh, sorry. At your left-hand side, that you can see. You can filter applications, which is important for you, product text. So product text is pretty much linked to this. Red map, whether you want to just to look on red map monoclonal antibody, very specific for recombinant antibodies, clonality, and so far and so on. There is a list of filter that you can filter. So this is the tag shown here. Product information, that's already well illustrated. This is the tested application and reactivity. We also have attached like ABCAM review. So there is a review from the researcher globally. Feel free to join us too and link to some publication. Last but not least, I just want to highlight that there's also a resource tab here to get those general protocol books that you need, and we even have like video training uploaded online. So this is an example of like a customer review around the world where you can get some of the information. Again, feel free to participate too. and share your passion on our website. Highlight again, that's all the guidebook, even troubleshooting tips to support our researchers and some of the articles related. So this brings us to the end of today's webinar. Thank you for joining us again. We kind of end early. At ABCAM, we remain committed to deliver high-quality products that accelerate and empower your research. So we look forward to support your scientific journey. And thank you.